and things have not been going well for me. So I'm hopeful that we'll be able to successfully do it this hour. There we go. Yeah, that should good. Leave that. And it picks up the microwave or the microwave microphone from over there. And we have our notes up. Okay, so hopefully, cross our fingers, this works. Oh, wait a minute. I had to screen share, didn't I? I'm still getting used to the system. This is very different from last year, the way I did it last year. I need to go here and share this screen. There. And then bring this up. And now that should be showing up over here. Okay. So hopefully we figured it out now. We got our volume. I did it fourth hour, but then the volume, I was trying to record the volume off of here, and apparently there's no microphone, even though it says microphone activated, but it's not working. So hopefully between the two computers, we can get it to work. I try to record all of the lessons for you guys so that if you're gone, or if you get home and you're like, how did she say to do that? You can pull that up, that video up. And, you know, I've got 4 tries at it for your class because I do this 4 times a day. So, all right. So we get started here. Introduction to chemistry is today. And we start out by talking about the 5 different branches of chemistry. The first branch is organic chemistry. Organic chemistry, when you think of organic, you probably think of going to the grocery store and having a section of the grocery store, particularly like in the meat section or in the produce and fruit section, that's organic, right? Well, in chemistry, it's a little different. In that section, what they mean is if it's meat, they haven't used any kind of medication like hormones or antibiotics or anything like that on the animal before it was dispatched. If it was fruits or vegetables, then it's typically we haven't used any herbicide or pesticides on this. Okay. In chemistry, it's different. In chemistry, organic means that these items have molecules in them that have hydrogen-carbon bonds, essentially. Okay. What that means is any living organism is going to be organic. Most of your herbicides and pesticides are actually organic. Okay, so it's very different than the one that you're used to. And it's a huge area of chemistry. If you get into college and you go into any kind of medical field, you'll generally have to take a course in organic chemistry. Inorganic chemistry is where we spend the majority of our time working with the other elements on the periodic table. We do work with hydrogen and carbon a little bit, but most of the stuff that we work with doesn't have hydrogen carbon bonds the same way as like the organic chemicals do. Analytical, if you've ever noticed, like on the side of food and stuff, they've got those nutrition labels. Those would all be from analytical chemists. So what they do is they get the product and then they analyze it, analytical, for what kind of substances make up that particular item. Physical chemistry has to do with thermodynamics and a few other things. If you take AP, we get into a few of those things in there. And then biochemistry is primarily what biology is about. Okay. If you go on and you take AP Bio, it will actually has a lot in there that you work with as far as biochemistry. The prerequisite here for AP Biology is not biology. It's actually chemistry. Because you can only cover the very surface of biology without understanding any of the chemistry behind it. It's kind of the building blocks or the foundation of biology. Scientific method, I'm guessing that you guys have probably spent a lot of time with the scientific method in the past when you were kids, younger, you've done a lot of that stuff. So we just kind of talk about it a little bit for a couple minutes and then we move on. Generally, it starts out with some sort of observation. That observation leads you to question what's going on. Then you make a guess, which we call a hypothesis in science, as to why this observable thing happened. That hypothesis typically is written as an if-then statement. Then you will do an experiment to test and see if that item actually worked the way you thought it would. You have data that you collect, and then you analyze that data. You look it over to see what kind of trends were present. And then finally, you have a conclusion. That conclusion answers whether the hypothesis was correct or not. If that hypothesis was correct, you might want to redo all of this again, just to make sure that you didn't have any errors. If it was incorrect, then you might want to go back and revise it and then do additional experimentation to try to solve that initial question. Okay, good. From this scientific method, then, you can end up establishing a theory or a scientific law. The theory, then, is going to explain some sort of phenomenon. So you're coming up with an idea that you think is correct. So, for example, if we were to look at the extinction of the dinosaurs, what caused the extinction of the dinosaurs? Okay, they have a theory out there that a big meteor came down and boom, sent up all kinds of ash into the environment that killed off the plants, and the plants died, then animals died, and then, the dinosaurs went with them. So that is an explanation. Do we know if that actually occurred? We got a lot of evidence to support it, but can we go back in time and actually see it happen? No. Okay. Scientific law then describes a phenomenon that will happen, and this you can observe. So, for example, if I lift this up and I let go, what's going to happen? It's going to fall. Okay. Universal gravitation law. Okay. We can see that happening. We have something that describes it, and we can observe it. Okay. So that is kind of the difference between a theory and a law. It is hot in here. Pull in some of that air. All right, good. That's good. Okay. So scientific law then is going to predict, and a theory is going to give you a logical explanation. So that's just kind of another way to phrase what I had on that Venn diagram. Okay, if you want to write it down, you can, but if you get the idea from the Venn diagram, it's good too. It's supposed to be the last hot day. It's supposed to cool off after day. Then we bring the fan over to the window and pull some air in from the outside. All right, good. Okay. So recording data, once we get our information, as we're doing our experiment, we have to record data as we go along. So the first type of data is qualitative. This is using your 5 senses. So how does the thing smell? What kind of sounds is it producing? Fizzing, popping, that sort of thing. Don't usually use taste in here very much, but if you're in the culinary world and then you're working on stuff, that one might come into play. Otherwise, it's primarily there's these four that we use in chemistry. Quantitative then is going to deal with numbers. So you've got some sort of quantity as opposed to some sort of quality. Okay. Come on. There we go. We also, when we're doing these experiments, we have independent variables and we have dependent variables. Okay. The dependent variable is going to depend on what you change about the independent variable. So if we change the amount of water, then the size of the plant is going to change. So the size of the plant depends on how you manipulate or change the amount of water. And if you can kind of think about the experiments in those terms, what depends on what, it'll help you to be able to identify how to place things on graphs. Okay? So graphs are going to have several different aspects, and I'm going to go through these on the next slide with you. I just kind of wanted to start here. You're also going to have homework tonight where you kind of go through this as well. So your graph is going to include a title. It's going to have your axes labeled, and those axes are going to be labeled using a concept called dry mix. I'll get into that on the next slide, what that means, because you're like, what are you talking about? You also have to have your units on there. You have to have your intervals, and they have to be equal, a legend, and then you have to have scales on each axis so that you're using the entire graph or as much of it as you possibly reasonably can do. Okay. All right. So I've got a blank slide here. Let's talk about how that's going to look. If I am given a graph, first of all, if I'm given this graph paper, I do not want a graph that is that big. Okay. I want it to take up as much in this direction and this direction as you reasonably can do. Okay. I don't want you dividing the thing out by the 0.22225, kind of thing to get it there. But reasonably, if you have a couple extra cells on each end or at the top, I'm okay with that. But trying to give me a graph that's this big when I've given you this much paper, you're going to get points off. All right. So we've got that established. When you do these, you need to make sure that you label your axes. So if we're talking about our plant, if we were discussing that, we talked about the plant height. And let's talk about the amount of water. Okay. The plant height depends upon the amount of water you give. So there is something called dry mix out there. This is how I remember what goes on what axis. The D is for dependent. And I didn't leave myself enough room here. Let's try this again. Responding. So it's responding. And it is going to go on the y-axis. This is responding to the manipulated. So what are you changing? That is your independent variable. And that is going to go on the x-axis. Okay. So when we're talking about our plant, we're changing the amount of water. So we are manipulating that. That means that it is going to go on my x-axis. So amount of water will go here. And my y-axis then is going to be plant height. Okay. So I've got these in the right spot. I also, with these labels, I need to include at the end in parentheses what unit I measured that in. So let's say that we measured the amount of water in milliliters, and we're measuring the height of the plant in centimeters. You get a point for this, a point for this, a point for this, and a point for this when I grade graphs. Okay. You need to make sure that your scales are right. So let's say that we started here with 0 and we're doing amount of water. If I have this as 10 milliliters and this is 20, I can't all of a sudden jump and put a 50 here. Okay. This spacing, each one of these is 10. This has to be what value? 30. Okay. You get the idea? Same thing going up this side. You lose points if you don't do that correctly. Then let's say that we're dealing with 2 different plants. Okay, so I need to put 2 lines on here. I need to put a legend on there. And I only have 1 pen color. I can't figure out yet how to get different pen colors up here. So let's say that this plant, I ended up with a graph like that. And this plant, I ended up with a graph like this. Okay. How could I differentiate between those? Well, I could have drawn this as a dotted line. I can do that now. Or I can take my points here, my data points, and I could mark each of those data points with a different, oops, this should be a star. I can mark each of those data points with a different thing to correspond to it. So these are dots. And then I need to provide my reader a legend. So I could go this, or I could put dashed lines, dots, solid line. This is plant A, and this is plant B. Okay, makes sense? So those are the types of things that I'm looking for when I'm grading your graphs in here. We always use line graphs in here. We never use anything else. Any questions on graphing? All right. So the next section of our notes is an I do. It's to get you kind of started on your homework. The I do for the first part here, they're independent and dependent variables, and they want us to identify those in our examples here. So we have the higher temperature of the air, the higher the temperature of the oven, the faster the cake will bake. So what depends on what? Right. So the speed of the cake baking, so speed cake will bake. Let's just do that. Speed cake will bake. That is dependent. So this is my dependent variable on the temperature of the air. So that would be my independent. This then is going to go on my x-axis, and this would go on my y. So lemon trees receiving the most water produce the most lemons. What depends on what? Right, the lemons produce depends. So the number of lemons, that is going to be my dependent variable. And then amount of water, that will be my independent. So if you just ask yourself what depends on what, and you identify those things, then you can easily place it in the graph. All right, respiration rate of the fish is determined by the water temperature. So what depends on what? Right. So this is going to be my respiration. So this is going to be my dependent variable, and the water temperature is going to be my independent. Easy enough? Okay. And then you can use your dry mix to figure out which one goes on which axis. All right. Next one, we're going to identify qualitative and quantitative. So take just 1 minute, fill that out, and then compare to the person next to you. Yeah, Elle, you can compare with the Tristans. You're now collectively known as the Tristans. Are you done? Easy enough? So find the ones that have numbers in them. Because remember, the numbers are your quantitative. So that's why we have N and N. And then here we're looking at qualities. So this is an L and L. Good? All right. So you have, I think we get out of here at 36. So you have about 10 minutes. Most people finished up in my other classes. You do have something that you want to go on the Chromebook to do to get this done. So if you haven't gotten into Google Classroom yet, you need to make sure that you get into my Google Classroom. Okay. I also updated the class calendar to include the calendar with links to different activities that we'll be doing in here. So here's the calendar. So here are the different activities. This is the same sheet that's in front of you right now in your packet, but I do have links to the different activities in case you're gone, you don't get a packet or something like that. Okay.